diffusion, because it's passive, if something is passive and doesn't require energy, you can't be making something more concentrated. Okay, you can only be making something less concentrated. All right, then we have facilitated diffusion. This is following the same premise that we're going down a gradient. Okay, it's also passive, which means it doesn't require energy. The difference is, in facilitated diffusion, whatever is moving down its gradient cannot cross the membrane on its own, right? And I think we talked about, well, I think it's coming later, things that can and can't cross the membrane. So in the case of facilitated diffusion, it doesn't require energy, but it needs a way to get across, and that way is a transport protein. Sometimes people call these channels. It's the same, they're talking about the same thing. Okay, and then in active transport, we require energy. So this implies if we're doing something, if we're doing work, okay, if something is not just going from high to low concentration, then we're moving it against its gradient. And so the energy that's often used in active transport, but not the only one, is ATP. And ATP is going to become a household name in here. If you're not familiar with it already, it's adenosine triphosphate. And when ATP is broken down by lysing off one of the three phosphates, going from triphosphate to diphosphate with the release of an inorganic phosphate, that releases energy. So there's a lot of energy tied up in the covalent bonds between the phosphates. The sweetest sound on earth, except you're drinking some energy toxin. Although you don't know what this is. Anyway,
$1 mystery toxin.
okay, so comparing the three, if we're going down a gradient, it's passive. If we're going up a gradient, it's active. If we're going down a gradient but require a protein to get it across the membrane, that's facilitated. Okay, and things that might require, things that would not require a protein, that would just be simple diffusion, would be things that have high permeability, like gases, usually. Ethanol, which is not a gas, but can move across membranes. Things that have moderate permeability would be molecules that have some polarity to them, like water. Water can cross membranes, but it doesn't go that easily. And actually, there are some channels that allow water to go faster when necessary. Things that would require some type of facilitated diffusion or some type of active transport would be things with low permeability. So moving sugars into a cell, for example, amino acids, proteins, ions, all of these things require help and sometimes energy. All right, I think that's where we ended. All right, okay. So let's keep going. A couple other things to remember, a couple terms. When we're thinking of gradients, gradients can come in more than one form. So a chemical gradient, like we see on the top, has more molecules on one side of the membrane than the other, but there's no charge involved. Okay, so chemical gradient would be differences in concentration across membranes, and then we will add our little disclaimer here: no charges involved. Okay, so glucose is a good example of this. Glucose is a neutral molecule. It's polar, and it's hydrophilic, so it doesn't cross the membrane easily, but it's not charged. Now, another theme that we'll see a lot, and it's going to play a really big role in the next big topic we do, which is aerobic respiration and the generation of energy, is an electrochemical gradient. This actually plays a role in lots of different things in the cells. So in an electrochemical gradient, there's a difference in concentration as well as a charge differential. So we can say, we'll use the term solid, difference in solid and charges across membranes. The example they have at the bottom there is a little hard to, it's not a great diagram, there's just too many things in there. But what they're trying to demonstrate is that there is a larger concentration of the purple spheres on the outside, which are meant to represent sodium. And sodium, because it's a charged molecule, if you have a lot of sodiums outside of the cell and few sodiums in, you're likely to have a charge differential. In this case, it would be a net positive charge on the outside. And so that charge differential across membranes is important for a lot of things. Actually, there's a table somewhere. Maybe down the road. Let's skip to it.
Dr. Payne,
Yes.
does that say charges or changes down there?
It could be either.
Charges, I
It's
think.
charges, it's just a bad R. Not my best word.
Thank
Not
you.
my worst, for sure. So here are some reasons why you might have electrochemical gradients. Let's just skip right to this and we'll go back and look at some examples of the molecules in a minute. So transporting ions, so we haven't seen these terms yet. I'm about to show you. Symporters and antiporters use hydrogen and sodium to take up nutrients and to export waste. So we'll see some examples of that. When the mitochondria and chloroplasts synthesize ATP, they're going to use hydrogen ion gradients, which we will often just call proton gradients. Regulating water on either side of the cell relies on gradients of charged molecules. Neuronal signaling and muscle contraction. These, of course, if you take anatomy and physiology, you'll spend a lot of time talking about sodium and potassium action potentials and transmitting signals between cells. And then won't really hit on this in here, but if you take microbiology, which you may if you're a biology or biology or biomedical major, bacteria also use proton gradients to swim. So the flagellum or flagella, plural, depending on the bacteria, use those gradients to rotate their little tail. Okay, let's go back. So the three things that, wait, have you done osmosis yet in the lab? You told me and I forgot. No, okay, because your exam is this week, isn't it?
Tomorrow.
You'll do great. You'll do great. What could go wrong? No, right. Okay, so let's think about osmosis. This will be an upcoming topic in your lab, but not this week, obviously. This week, focus on the exam. For osmosis, this is the net movement of water across membranes as a result of solute concentrations. The net movement of water across membranes as a result of solute concentrations. And so whether or not water moves back and forth across the membrane will depend on which side of the membrane there's a higher solute concentration. Or in some cases, of course, they can be equal. So let's think of our three possibilities here. The first possibility, and when we talk about these terms, oops, where am I drawing? Isotonic, hypertonic, and hypotonic, we are referring to the solution that the cell is in. So let's say that we have our giant pet cell. Hey, buddy. And he's swimming around in this little beaker. And the solute concentration, it could be anything. Let's say it's sodium. Well, actually, it doesn't matter. We don't have to say what it is. Let's just say it's a solid. If the concentration of the solid on the inside of the cell is the same as the concentration on the outside of the cell, this is a situation where the solution is said to be isotonic. Iso meaning same, right? And so the solution is isotonic. There's no net movement of water into or out of the cell. Okay, and when we say net movement, this is a term that I won't assume you know, but maybe you know it. When there's no net movement, that means that water may be going into the cell, water may be going out of the cell, but overall, you break even. So for every water molecule that comes in, one goes out. So it doesn't mean water isn't moving, it just means there's no difference when it's all said and done. Okay, so your cell's pretty happy in that little solution. Now let's say that you have another pet cell, and this pet cell is in a solution that is hypertonic. Hyper meaning excess or more than. So if this solution is hypertonic, that means that we have more solutes in the solution than in the cell. Okay, so I'll exaggerate it there and not have any in the cell. So if the solution is hypertonic, remember osmosis is the net movement of water in response or as a result of the solute concentrations. Water is always going to go where the solute is at highest concentration. So the net movement of water is going to be out of the cell. Okay? So in this situation, the cell will lose water and the cell will probably shrink, okay, especially if it's an animal cell. All right, the third option, I'll put it over here, hypotonic. So here's your third pet cell. If the solution is hypotonic, that means that we have less than, right? Hypo, the opposite of hyper, so hypotonic means less solutes, and since we're talking about the solution, that means our cell is going to have more, we can put a couple solids out there. Our cell is going to have a higher concentration of solids than the solution that surrounds it. Okay, and so remember, water moves in the direction of the highest concentration of solid. So in this case, whoops, we'll say net movement of water into the cell. And so the consequence of this will be that the cell might enlarge or swell up. Okay? And so by changing the solid concentration, you can alter what's happening in the cell. So this comes in handy in a lot of practical uses. For one thing, well, I think there's a picture of blood cells. Yeah, there's a creepy picture of blood cells. So we do not want the cells, of course, to shrink. We don't want the cells to burst. So if you, as a practical example, go get fluids at a hospital, let's say you're dehydrated or you're having some type of surgery and they're trying to keep your fluid levels high. They're not just pumping water into your body, because if they pump water into your body, your cells are going to swell up, and that's dangerous. And they're not pumping super salty solutions into your body either, because you don't want your cells to shrink. And so the saline solution that they put is going to be perfectly concentrated with solid, so that it makes your cells happy, and that fluid, as it goes into your body, is absorbed appropriately. So you can't just put distilled water, for example. Now, there's a different effect when we think about plant cells. Plant cells have a constraint that our cells don't have, which is that they have cell walls. So this changes the dynamic of what happens in hypertonic or hypotonic solutions, because if it is in a solution that is hypertonic, remember, hypertonic means that movement of water is out of the cell. If water goes out of the cell wall doesn't actually change structure, right? The cell wall is a rigid structure. So the membrane of the cell shrinks and pulls away from the cell wall, but it has to stay inside that chamber. But if you put it in a hypotonic solution where the net movement is into the cell is going to swell, but again, the cell wall is going to constrain that the growth of that cell in size, which means that even though it swells up, it doesn't get that much bigger. Instead, it pushes against the wall. So this is the difference of a cell or a plant that would be, upright, and a plant that would be wilted. Now, one of the places that you see this is in aquatic plants. Because in aquatic plants, of course, they're surrounded by a solution of water, right? And so if that solution of water is hypo or hypertonic, those plant cells will adjust accordingly. But a place that you see it that is not as obvious, because you can't put it under a microscope, would be on the roots of plants, like garden plants, for example, would be an example. So if you over-fertilize a garden, one of the things that happens is you're putting a lot of solute around those plants. And so if the concentration of solute is much higher outside of the plants than inside of the plants, water is going to move out of the cell. So you can actually dehydrate your plants by fertilizing, because all of those, mostly ions and different chemical compounds, are pulling water out of the plants as water moves in the direction of the higher solid concentration. So you have to strike a balance. So life is all about, right? Balance. Who's living a balanced life? All right, good. 2 of you. That's not bad. We're 1/30th of the way. Speaking of imbalance, all of my classes this semester have a disproportionately lower than average number of left-handed people. It's a trend. I don't know where they've gone. It's the lowest percentage I've ever had. It's got me concerned. Okay, where were we? Oh yeah, channels. So in order to move solids, or in order to move chemicals into or out of the cell, we talked about how things can facilitate that. So this could be an example of facilitated diffusion. If you notice, things are moving along their gradient. But facilitated diffusion doesn't just mean that a channel is open all the time. So we can have things that open and close, and channels like this are called gated channels, like just exactly what the name implies. And different things can open or close those channels in response to things that the cell needs. One of the important terms that's associated with this section is conformational change. And let's see, they define it up here. Undergoes a conformational change. No, they don't define it. That's okay. So the terms conformational change something's conformation is its shape. Okay, it's not a term we use outside of biology classrooms too often, but that's what it means. The conformation is the shape. And so a conformational change is essentially a shape change. And this usually carries with it some consequence. So this is a structural shift in the shape of a protein that has a biological consequence. And this happens so often in biological systems. My biochemistry teacher in college, I remember, joked that if you don't know the answer to a question just right, it undergoes a conformational change. And so many times in biochemistry, that will be the truth. So the example they give here is that the conformational change that's taking place is in this membrane transport protein, okay? And so a membrane transport protein, remember, is trying to move a molecule across the membrane that can't get across on its own. So whatever this molecule is supposed to be, it must have a charge or just be too big or whatever. But it can't get across on its own. And so in order to get across, it goes into this little pocket in the protein. Some people call it a cavity. And this cavity is hydrophilic. So that means the amino acids that are in this inner part of this protein have hydrophilic or polar side chains, right? Whereas the amino acids that form the outside of this protein must be hydrophobic because they're interacting with the phospholipids, all right? And so the conformational change takes place when this solute hides inside the hydrophilic pocket and something triggers that protein to go from outside to inside. Okay, it undergoes a structural change, and now this solute is released to the inside of the cell. So this may or may not require energy. Sometimes it does, sometimes it doesn't, depending on the protein we're talking about. But it's a conformational shape change that tricks, well, it doesn't trick anything, but it allows this solute to cross a membrane that it otherwise would not be able to cross. And depending on which direction it's going and how many molecules are going with it, we have 3 different possibilities. I won't write them out there because they're defined very easily here on the board. But let's look at the first one. This would be like the example we just saw in the previous picture. This is a uniporter, right? These are all plays on the word transporter. So a uniporter, uni meaning one, moves a single solid in one direction. Okay, so whatever that solid may be, it's the only one that this transport protein facilitates the movement of. And it's only going in one direction, meaning that it's only going in this example. Okay, the second one is called a symporter. You probably see the prefix sym, like symbiosis, like working together. So a symporter is a transport protein that moves 2 solids, and they're both going in the same direction. So they can either both go in or they can both go out, but they're moving as a pair. We're going to look at, well, actually, we'll look at examples of the third one, not the second one. So uni, 1 solid, 1 direction. Symporter, 2 solids in the same direction. And then the antiporter, this one's interesting. It's a dual purpose protein. It allows one molecule to go in and it allows a different molecule to go out. And so this is effective at making electrochemical gradients because you can move a molecule with 1 charge in and a molecule with the opposite charge out, and you can really stack the deck in your favor. So that's an antiporter. So uni, symporter, antiporter. And this isn't implying that these are facilitated diffusion or active transport. They can, they're examples of both of those types of things that are uni, symporter, and antiporters. Okay, so here is an example of 2 types of active transport that are using proteins and energy. The first one is primary active transport. So prior to this, we defined active transport as movement of solids across a membrane against their gradient and utilizing energy. So now we're going to be a little more specific and look at types of active transport. So we already are implying that energy is involved by the fact that we're saying active. The first one is called primary active transport. And primary active transport, again, it's implied that something is moving against its gradient, so I'm not going to use that in the definition. It's implied that energy is used. But the fact that it's primary means the transporter utilizes chemical energy, and I don't think we'll see an example that isn't ATP. Oh, wait, I don't think so. So I'm just going to put ATP as our example in the definition. The transporter utilizes chemical energy to move solids. I guess I'll go ahead and put the whole definition against their gradient. All right, and remember ATP splitting into ADP plus inorganic phosphate releases energy when that phosphate pops off, and that energy can be used to do work. In this case, it's moving hydrogen ions, also known as protons, outside of, or to the other side of the cell to create a gradient. Okay, so now we have a gradient of 1 solid, and that was directly the result of using chemical energy in the form of ATP. And one thing that the cell could do that's kind of curious is it can use that gradient to do other work. So it spends energy to create a gradient, and then that gradient wants to resolve itself, right? If 2 things are out of equilibrium, they want to get back to equilibrium. So the protons want to come back into the cell. If you provide them a way to do so, then they will. So one molecule is busy making the gradient, another transport protein is busy allowing hydrogen to come back to equilibrium. So when hydrogen does that, the movement of those hydrogen ions drives a conformational change. Makes a little sound when it happens. And that conformational change moves another solute along with the proton. All right, so what is that red one supposed to be? Sucrose. So sucrose is really just along for the ride here. And so by using the hydrogen gradient, you get the benefit of also moving sucrose against its gradient. So hydrogen is coming down its gradient, sucrose is going against its gradient. All right, let's see if we can say that more concisely. So secondary active transport. Think in my head before I start writing it. Okay. Utilizes the gradient created by primary active transport
Thank you.
utilizes the gradient created by primary active transport to move a different solute against its gradient. Utilizes the gradient created by primary active transport. Actually, a better way to say it, sorry if you're using pen, created by primary active transport of one solid to move a different solid against its gradient. Best way to understand this is to look at that diagram. That diagram is really helpful. Wonder what osmotic catastrophe would happen if you put an energy drink in your IV. Reminds me of the Evanescence song, Bring Me to Life. I'm definitely anti-energy drink for a number of reasons. One, because who knows what that's doing to your body. But I had a student that stayed up all night to study for an exam, and they had never had an energy drink before. And they were on the verge of, they were at their body's limit. And so they came to the exam and they drank, maybe they'd already had one energy drink and they were on their second. And they just, they just freaked out. They couldn't move, they couldn't write. They said they couldn't see the exam anymore. They said at some point, I could no longer read the words. It just blew their brain up. That's what I think about when I see someone drink an energy drink, that poor student. Same thing happened to a student that had their wisdom teeth out and then ate a piece of cake after 2 days without food and a lot of pain meds. Different bad idea, but still a bad idea. Don't make cake the first thing you go for after mashed potatoes. Okay, so let's keep these concepts in mind as we look now at a very specific example in some detail. I don't want you to think that this is the only type of active transport. It is just one very well-studied example that we know a lot of details about. So we're going to use it to understand what's happening. Okay, so this is called the sodium-potassium ATPase. That's the name of the pump. And the name of this pump has in its nomenclature the word ATPase. So this means that this pump is going to be enzymatic, right? The ACE means it's an enzyme. An ATPase, by definition, not just because of this pump, but whenever we see something that's an ATPase, what that means is it is going to take ATP, break off one of the phosphates, and release energy in the process, right? And so this, let's see, this reaction, remember the little I stands for inorganic, it's not pi. This reaction is called this is what I'll always call it from this point forward, the hydrolysis of ATP. Because water is involved in lysing that phosphate. So hydrolysis of ATP, and I'll put parenthetically, releases energy. So an ATPase catalyzes the hydrolysis of ATP. Now in this case, that's not the only thing that it does. And this is probably a good time to say this about enzymes. Enzymes can do more than one thing. So sometimes an enzyme will have multiple different reactions that it catalyzes. And each of those reactions we call an activity. So we might say that this enzyme has ATPase activity. That means that one of the things it catalyzes is the breakdown of ATP. But it also might have some other type of activity. So in addition to breaking down ATP, one of the activities this enzyme has is to pump ions back and forth across the membrane. Okay? So sodium, potassium, ATPase, let's give it a little short definition before we dive in. This enzyme is going to move 3 sodium ions out, comma, and 2 potassium ions into the cell. Actually, let me say this a better way. So moves 3 sodium ions out and 2 potassium ions into the cell with the hydrolysis, that's an R, of ATP. Okay, so if you give a single ATP molecule to this pump, it moves 3 sodiums out, 2 potassiums in. All right, so that's how much you get for each molecule of ATP. So if this cell had 2 molecules of ATP at its disposal, we could move 6 sodiums out and 4 potassiums in, and so on. All right, and the overall goal will be to create this gradient where we have, remember, the brackets mean concentration, where we have a high concentration of sodium out and a low concentration of potassium on the outside, and a low concentration of sodium on the inside and a high concentration of potassium. So if we're creating a gradient, we're moving molecules against their normal equilibrium will and putting them in sections of regions of high concentration. Okay, this is a stepwise procedure, so there are multiple stages through which this goes. Actually, they're annotated on this one, but that's a lot of, I don't know, I may annotate it differently. And so we think of this process going from the left of the screen to the right, which is the different things that this single molecule is doing. It's not 4 different molecules or transport proteins, it's one going through different stages. We think of this as kind of a cycle, meaning one cycle of this pump utilizes 1 ATP, and as we said before, moves 3 sodiums out and 2 potassiums in. So that's one cycle of the pump. And so during each cycle of the pump, a number of things are going to happen. And just a couple things to clarify what we're looking at here. E1 is referring to, the E refers to the enzyme. So this is shape or conformation 1, and this is conformation 2. Oh, this also. Okay, and then it goes back, if you notice, to E1. All right, so let's put our stages. Now that we've defined what this pump is and what it does, let's talk about how it does it. Okay, so the first thing that happens is that 3 sodium ions bind the E1, we'll say structure. Actually, I'm going to say form. So this, again, is a reference to the protein. 3 sodium ions bind the E1 form on the intracellular side. Okay, so we're facing in. So 3 sodiums bind. That means that because sodiums are charged ions, these must be hydrophilic pockets. Okay, so those sodium ions bind on the inside. And the second thing that happens is that ATP is hydrolyzed, and this doesn't always happen when ATP is hydrolyzed, but in this particular instance, the purpose of lobbing off that phosphate is to actually attach it to the protein. So sometimes the phosphate is just released. It's not used for anything other than the energy production. But in this case, the phosphate's actually added. So ATP is hydrolyzed. The phosphate is attached to the pump. So we're calling the sodium potassium ATPase the pump. And when you add a phosphate, keep in mind a phosphate is a charged functional group with 4 oxygens and a phosphorus. It's a pretty big molecule, okay? And so it's actually going to have an effect on the structure of that protein. And if you notice, it flips it to the outside. So we know what the change in the shape of a protein is called. So the ATP is hydrolyzed, the phosphate is attached to the pump. The pump undergoes what? A what? Give you a hint. Yes. Nice. It was getting hard too. It undergoes a conformational change. I've run out of real estate. I'll move over here. This is continuation of what I was writing there. Undergoes a conformational change to the E2 form, releasing 3 sodium ions to the outside of the cell. So as long as that phosphate is attached, our protein is going to be outward facing in the E2 form because of its conformational change. All right, now what happens next? When it faces out, because it has a different structure, the sodium no longer has affinity. So these binding sites for sodium are altered in the conformational change, and now it doesn't have any interest in them anymore, right? That's yesterday's news. And so in its new shape, though, it has high affinity for potassium ions. So the sodiums are released. What number are we on? 3. And we'll say this, in the E2 form, the pump now has high affinity for 2 potassium ions. All right, it has a potassium-shaped hole in its heart. 2 potassium-shaped holes. And so, because it has that, it will bind to potassium ions, and the binding of those potassium ions triggers the release of the phosphate. Okay, in the E2 form, the pump now has high affinity potassium ions, which bind and cause release of the phosphate. Goodbye, Norma Jean. Now, what do we know about this protein when it's not phosphorylated? I'll go back to its E1 shape. Okay, so now the pump returns to the E1 form, releasing 2 potassium ions into the cell. And then we begin again. Probably the best song on Red. Questions? How we doing? Energy drinks kicking in? No. What's in that? I don't need to know. Whatever gets you through the day, man. Okay, let's see if it says anything here that we didn't say. Sodium binds from the cytosol, ATP is hydrolyzed, ADP is released, and phosphate is covalently attached to the pump, switching to the E2. Okay. E2, remember, switches out. Sodium are released outside of the cell. 2 potassium bind from outside the cell, which triggers the release of the phosphates. The pump switches to E1, 2 phosphate are released, the process repeats. Okay, I think we covered all our bases. I felt like there was something I was about to say. Oh, I know what I was going to say. The process of adding a phosphate, I think I might have said it as I was moving along, but I've never deliberately described it. This process is called phosphorylation. So we'll talk about that a lot this semester. If we're putting a phosphate on something, we can say that that thing is now phosphorylated, or we can say that this is the process of phosphorylation. That's what we're talking about. That may be completely straightforward, I don't know. Doesn't hurt to ask or to say. Okay, we've already described why we might be doing these things. Okay, so that's transport of small molecules and ions. One type of transport that I think, can I erase this too? One type of transport I think we talked about very briefly, maybe, did we talk about it during the cell anatomy section, endocytosis and exocytosis? Maybe we did. Okay, well, either way, it belongs here. And so endocytosis and exocytosis are the movement of large volumes of solids into and out of the cell. Okay, so let's describe it like this. Transport of large volumes of solids or large structures. So you could, for example, transport bacterial cells into a cell, into a eukaryotic cell. All right, so that being said, if we are doing exocytosis, is that the first one they list here? Exocytosis, this is movement. Actually, we can use one way that we can, a word that's a catch-all for the movement of, or not the movement of, but for solids or large structures, we can call these collectively cargo. It's the thing we're moving, okay? So exocytosis is movement of cargo out of the cell. And the way that we're going to move these things out of the cell is in vesicles. Okay, so the movement of cargo out of the cell in vesicles. And some examples that they give, hormones are exocytosed. So if you're making insulin or glucagon in your pancreas and you're trying to regulate your blood sugar, those are going to be exocytosed. If you have digestive enzymes that you're releasing into your intestine to digest, of course, your meal, that's going to be exocytosis. And let's take a look at that before we move on to the other half. And so exocytosis is usually going to involve the Golgi apparatus, right? We describe the Golgi apparatus as the organelle that ships things to their final destination. In this case, the final destination is out of the cell. And so our Golgi apparatus has packaged whatever that cargo is, okay, into this vesicle. And one of the ways that you package something into a vesicle involves proteins that give that vesicle a circular spherical shape, right? The phospholipids themselves have no rigid structural capacity. And so if we wrap this up with this protein coat, then it will form this nice spherical shape, and it will travel to the plasma membrane. And remember, the membrane of our organelles is composed in more or less, this isn't 100% true, but it's composed with very similar phospholipid content as the plasma membrane, which means they can fuse. Can fuse, not they confuse, like they confuse us. They can fuse. And so if we shed those coat proteins, those phospholipids can blend together just like two soap bubbles could fuse. And when they fuse, that means that the inside of this vesicle is now the exterior of the cell, okay? So when it fuses with the plasma membrane, those solids leave the cell. Now, the other type of large volume transport is endocytosis. And this is, as you would guess, movement of cargo into the cell. Hello. Also using vesicles. And basically, this is the opposite of exocytosis in a very literal sense, because the membrane folds in. Well, let's take a look at the picture. The membrane folds in, which is called invagination. Proteins that are coat proteins coat the vesicle, giving it that shape. And then that vesicle travels somewhere into the cell. Now, usually, if we're bringing stuff into the cell, we're either going to break it down to destroy it, or we're going to break it down to use it as a nutrient. But either way, if we're breaking something down, a lysosome is probably going to be involved. Okay, lysosomes, remember, are the organelles that degrade and break down molecules and compounds in the cell. Now, one of the things that this sometimes involves is a receptor that grabs onto that particle before the invagination occurs. Okay, and depending on what type of thing we're bringing in, we can subcategorize this into two groups: pinocytosis and phagocytosis. So pinocytosis is bringing in fluid or liquids. Okay, so there may be something in the environment that is maybe nutrient rich, and you're going to bring it in and large volumes of liquid. So this is, let's see. No, let me say it a different way. We can just say endocytosis of liquids or fluid. The more exciting one is phagocytosis. And phagocytosis, well, let's look at our chart of examples. Phagocytosis is when cells, especially immune system cells, engulf and destroy large pathogens like bacteria. So phagocytosis is endocytosis of other cells or large solids. And the cells that bring in these large solids or other cells like bacterial cells are called phagocytes. So this is maybe an extension of this. The cells that do it, if you're curious, are called phagocytes. Cytes is a root for cell, and phago is a root for engulfing or eating. Okay? So one of the most common cells that does this is called a macrophage, which means a large phagocytic cell. And these are cells in your immune system that engulf viruses, bacteria, things that you are trying to remove from your body. They also engulf some of your cells. So for example, a macrophage in your body might swallow up some fragments of a cell that has undergone apoptosis, which is when a cell implodes. Basically, some people call it cellular suicide. And so some of your cells, if they have too many mutations, for example, break themselves down. Instead of risking becoming a tumor, they just go ahead and undergo this cellular suicide, and then phagocytes like macrophages come up and gobble up the pieces, and then problem solved. All right, let's see what else we have. Oh, let's see. Let's stop there. Let's not start a new topic with 10 minutes.